It's my pleasure to introduce tonight's lecturer, Dr. Robyn Lindley, who is an Honorary Senior Fellow in the Department of Pathology in our faculty, and she's also the Chief Scientific Officer and Director of Gene Mutation Diagnostic Company, GMDX Co. Proprietary Limited. With first degrees in physics and informatics, Robyn moved into immunogenetics and has published on the molecular mechanisms of evolution and somatic hypermutation in the immune system for almost 2 decades. It's my pleasure to welcome Robyn to give her lecture tonight, the R.D. Wright lecture, The Hunt for the Source of Mutations in Cancer, a story of discovery and innovation at the coalface.
I'll open by making a couple of comments about what's happening globally with genomics at the moment. Everyone's aware that there's a lot of development, a lot of investment in genomic research. And certainly there are a lot of new findings. These findings are expected to actually make a very fundamental difference to our lives, but not only our lives, how we perceive ourselves, how we view ourselves, and how we think of disease. Most of us know that the human genome consists of a basic program, really, if you look at it from the analytical point of view. It just consists of A, G, C's and T's and sequences. Yet we still know so little about the rules governing how these little programs actually interact with each other. We have a lot of work to do, and I expect that it's probably going to be another 20, even 50 years before we unfathom all the secrets. And one of the amazing facts about this little genome is that it actually contains less information than the last Microsoft Works program you actually downloaded onto your computer. When you think about it in those terms, we probably realise that there are a lot more rules that we've yet to understand. Professor Hargraves, Dean of the Faculty of Medicine, Dentistry and Health Sciences, faculty staff, ladies and gentlemen, and friends of the Sir Roy Douglas Wright, thank you. This evening is my privilege to describe to you just a very few of the new rules we're uncovering about how the genome actually functions and what their relationship is with disease. What I hope for each of you is that you actually have a few bits of extra information that will actually fundamentally alter the way you consider cancer and perhaps the way you think about yourselves and your life as you actually, all of us, undergo mutational change as we age, and unfortunately some of us faster than others. I'll begin by starting off with a brief background of where we've been in our thinking about cancer and our genes. And forgive me for those people who understand the science, but I just want to also spend a couple of minutes conveying 3 key concepts so that everyone here can hopefully walk out of the room and have some understanding of what the rules and what the hell I'm talking about when I'm explaining some of the new diagnostics that we're planning to introduce. The first thing is, it's very simple. Previous dogma, cancer's caused by random mutations. It wasn't so long ago that that was what was actually expressed. We now know that cancer is caused by genomes. We know that it's caused by exogenous factors in the environment. We know that UV light, too much, and we might get malignant melanoma. We also know now that it's caused by endogenous factors. They're the natural enzymes and proteins, if you like, that are actually part of our immune system and perform other functions. But when things go wrong, as I'll explain in some of the subsequent slides, that's when we're prone to develop cancer. So let's start with the first of 3 concepts, and forgive me for the science. First concept is really about what a motif is. What do we mean by motif? A motif is simply a short nucleotide sequence that surrounds any particular mutation that we analyse. The second concept is deaminase, and this is a word you'll hear frequently throughout the rest of the presentation. A deaminase is a protein that actually is part of our innate immune response to viral infection, bacterial infection, and how it operates is by clipping on to DNA or RNA at a particular motif, and we've just defined motif, makes a chemical change to one of the nucleotides it wishes to mutate, and then that mutation gives rise to a new nucleotide in that site. There are two families of deaminase. The first deaminase family is made of cytidine deaminases. They're the ones that all they do is simply mutate Cs. We record that as G mutations when we're looking at mutation tables, simply because Gs and Cs are opposite each other, but nature's found a ways of introducing Gs and C mutations because DNA is double-stranded. It's the complementarity of our DNA and our nucleotide sequence. The other family is called the adenosine deaminases. They're the ones that deaminate and then change an A nucleotide into something else. And an A, we know from basic science, is opposite a T. So we don't need a T deaminase, we just have A's, and again, because of the complementarity of strands, we end up, as a result, through cytidine and adenosine deaminases, we end up with mutations of A's, G's, C's and T's, wherever these deaminases may choose to, or perhaps be directed, because we don't understand all the rules yet, to cause mutations in our genome. The third concept, before we proceed, is really about codon context. Before our genes can make protein to produce the new cells, to make us function better, to replace old ones that are perhaps worn out, we actually have to have what's called nucleic acid sequence, and they're little triplet codes, if you like, or 3 nucleotides. And what we're going to talk about will show the relevance of those. In other words, you change one nucleotide in that trinucleotide sequence of a codon, then you change the protein that is produced. But not in all cases, but we won't go into the minutiae and the detail. But we really need to understand at this point that codon context, when we're talking for sites of mutations, is very important. And the mutation can occur at either the first, second or third nucleotide in a codon when we read the genome from 5' to 3', and that's just biology talk for the direction of transcription when DNA or RNA is being copied. I'd like to now just go back and spend a couple of minutes talking about where we've been conceptually in terms of understanding the deaminase activity. Take you back to 2004. In 2004, we had just discovered in 2000 that AID, one of the main deaminases, was discovered. We knew that there were some ADARs. We didn't know their function. But there were actually 2 studies that showed that perhaps after all, mutations on our genes are not so random as previously thought. They found some associations between the near neighbour nucleotides and where the mutation actually occurred. But unfortunately, they also looked at the different percentages of the nucleotides surrounding each of the mutations, and they described the methods as variously as sloppy mechanisms and didn't believe they were really, really specific. But they were the first to find the association and the fact that they were not random. And there's some data by Beale, there's also some by Pham, and then there was actually a huge gap in the literature where that science wasn't progressed. I then bring you to 2006, where with some colleagues from the Australian National University, we did some bioinformatics research, and remember that's the stage where we started to amass huge amounts of genetic information, and mutational databases on cancer were actually starting to really get some data that you could get your hands on and perhaps make some clinical sense out of it. What we found was the evidence of mRNA in between. In other words, that part of the process of mutation involved an mRNA. We now know that the adenosine deaminases actually target messenger RNA, if you like, that is then copied and made into new DNA. We call the process of mRNA to DNA reverse transcription. There have also been discoveries of reverse transcriptase, which is Polita, which is the only known reverse transcriptase to act in this setting. And so you see there's an information loop emerging, and we're starting to build a knowledge of exactly how or what opportunity the deaminases may have to mutate the gene. The next step occurred in 210, and this is one of the most significant ones for me in terms of changing or altering my thinking about how mutations occur and what the source might be. In 210, we actually, and you know, anecdotally, it was actually a 48-degree day in Western Australia, and a long weekend, we went into the lab and started looking at the data and asking new questions. We looked at the IgG genes, and they're the immunoglobulin genes, if you like, variable region genes that our immune system's based on. And we plotted out all the A to T's, A to G's, A to C's, and put them in the tables you see behind me. We then compared that to some cancer data that was also readily publicly available. And the example I've shown there is lung small cell carcinoma. We looked at 6 other cancers at the same time and plotted the same tables, and the striking similarity between what was happening in the immunoglobulin genes and the cancer patterns that occurred across all cancers that we looked at was striking. And it really, you know, asked us to ask some new questions. And what we mean by strand bias, because this has other implications as well, when you see there that there are more A than T, it's obvious that if the mutations of A and T are random, you'd expect equal numbers. But in all of the cancers we looked at, there were far more mutations of A than Ts. We call that strand bias again because A is opposite of T in that, you know, the Watson and Crick complement of our double-stranded DNA. And there's also ratios of Gs to Cs as well, which didn't make sense unless there was some sort of causal, same cause actually resulting in these patterns. The other thing at the time that was influencing, I guess, my thinking at least, was the genome is a very fluid entity. After having looked at evolutionary evidence for acquired inheritance, looking at the immune system functions and looking at developmental processes, the gene is incredibly fluid. It's a living entity within all of us, and it is influenced by what we do, and we're influenced by what we actually, you know, inherit from our parents. And if you look at it from an information flow, you see that to have that flow of information and even forgetting the evolutionary implications, just purely from the point of view of intercellular, you know, intergenerational production of cells from one generation to the next, there are a number of steps that really need to ensure that that information is able to flow. The key question at this point arose: how can we show that mutations are caused by deaminases? We already knew some of the deaminases were there. There was some speculation by Honjo from Japan, for example, Neuberger in the UK. There were a few people speculating that deaminases cause cancer, but no one actually had shown the association, and no one knew actually how to go about doing it at that stage. But certainly there was some emerging evidence that this probably is the case. So I guess my question is, what causes mutations? How do we identify these deaminases? So the next part of the talk is, the path identifying the source of mutations. The first thing I did was look for a database. Because I was doing it manually, there were no software tools for me. I looked everywhere to find some quick leg ups in terms of data analysis. There were none. I actually went right back to first principles and looked at the TP53 gene. A lot of knowledge was already built around understanding the functionality of the TP53 gene. It's not a large gene. There were lots of cancer mutation available in the IARC database that I could work with for the IARC gene, so I set about looking at those manually. Now, the first pattern that was observed was that the mutations land, whether they are Cs or Gs, and in biology speak, that means the transcribed strand or the non-transcribed strand. And they target a particular nucleotide within the codon context. What was also obvious when you look at the AID, which is one of the deaminases you might recall, that is the motif. It depended on the motif. It also depended on the point in the codon structure that the mutation occurred. And you can see that it's statistically significant. There's a very clear evidence that that might be the case. And this is just working with a trinucleotide motif, I might add. The next question was, is this pattern observed for other deaminases, given what we knew about the deaminases at that point in time and some of the motifs that were starting to emerge, whether they're really accurate or not, they still showed the same pattern. And that, when I say really accurate, I meant dinucleotide sequence is not that accurate, yet the results and the pattern were clearly very strong. The other thing that started to emerge from these analyses were that there's some potential clinical implications for the data we're looking at, because some of the cancers we looked at actually showed a very high, or far higher than expected, number of mutations due to ApoBec3G, for example. We knew that 3G is one of the first lines of defence against viral or retroviral infection. And when we looked at cervical cancer, for example, every single sample we looked at, as well as the IARC TP53 database, pooled cervical cancer data, the same pattern kept emerging over and over again. So I have no doubt about the repeatability of the pattern across genes, and looking at it from the different cancer point of views, we can see the different numbers of mutations emerging associated with a particular motif and a particular site within the codon context, if you like. The other thing we looked at was, what's the difference if we plot on the horizontal lines some of these deaminase motifs by codon and motif, if you like, codon site and motif, against the relative number of mutations for particular cancers. You see that we start to see the differentiation occurring, very different patterns across the different cancers we looked at. I've just put 4 examples there so you could see visually. And what it means is, and this is in agreement with what had previously been found by other people, that deaminases, most of them are tissue or tissue group specific. There is no such thing as a single set of deaminases that act on all tissue. Of course, there are a group of deaminases that are ubiquitous in the sense that they are involved in fighting viruses or playing their role in a number of tissues. The next thing we looked at was individual samples. Even within the group of cancers we looked at, a particular cohort, for example, bladder cancer, you break it down to individuals, you see more variation. In other words, there's a lot of variation among individuals with the type of deaminases that are the likely cause of their mutations and their mutation pattern. And this is more recent work now, you see, but the deaminases, the specificity of the deaminases increases dramatically when, and now that we've got the software tools, we're able to do this, and we're able to find that the specificity increases incredibly to the point of around 5 or 6 nucleotides to define a motif, which makes it even more powerful in terms of identifying which nucleotide, which deaminase we're actually looking at or is responsible for a particular set of mutations. The other thing we looked at or found more recently is, and this is consistent with what the work of others as well, they're finding that at some point in oncogenic pathways, there is a point where metastasis is triggered, where recurrence is predicted, in other words, or recurrence or progression of disease. There's also some work showing that some of the deaminases, and this is in vivo and in vitro work, that some of these deaminases, they're not all cancer-causing or cancer-promoting, tumour-promoting, if you like. Some of them are actually tumour suppressors. In other words, they're the good ones that you want. So we set about analysing some data to see if we could tease out which ones actually predict cancer. And we were fortunate enough to have a data set that actually had the right clinical annotations and also the data from apparently healthy tissue and tumour tissue, and we just looked at the different mutations that gave rise to the tumour compared to the healthy tissue for each individual, we found a number of motifs, if you like, that were targeted specifically to a particular codon site that were actually associated strongly with progression. In other words, if we developed a panel of them, the question was, could we use these as a prognostic predictor of recurrence or progression of disease? We did that with the samples we had and found out there was a very strong association, and the Kaplan-Meier plot you see there shows the sensitivity at 95%. We have much more work to do in this. This is very recent work. We need data that are consistent. We know that some of the platforms use different data pipelines. We know that the different methods used to collect samples. We believe that we can see it in the data. We can also see it in other cancer data sets. These deaminases, at some point, something triggers them to change their binding patterns, if you like, and that is associated with prediction of disease or metastases. There's also some other work, obviously, in the cell biology, cell surface areas, that have actually shown these changes occurring as well. The most significant finding recently that brings deaminases and targeted somatic mutation, if you like, when we've got a motif and we've got a codon site, into the fore is that there's a remarkable similarity between the ovarian cancer data that I just showed you the Kaplan-Meier plot for and the IG, or the mouse passenger transgene, the V-Kappa-Rox gene. The V-Kappa-Rox gene undergoes somatic hypermutation in the immunoglobulin genes. It does not undergo selection, so it's probably the best representation of what we can call somatic hypermutation pattern if we look at it from the point of view of the deaminase source of each of the mutations. When we compare using AID motif and using the sites for the codon, you can see that there's an incredible similarity between what's observed in ovarian cancer and what's observed in the mouse Kappa-Rox gene. The question next is this the same for ADARs? And yes, again, a striking similarity. And this is 2006 work, even though the data has been sitting around for many years. So that brings me to defining targeted somatic mutation with these new phenomena. Targeted somatic mutation is a somatic hypermutation-like process. In other words, it's like the process that's been studied for 60 years by immunologists in the immunoglobulin genes. It's very, very similar, only it happens in our other tissues, other genes. It's a universal process that happens on all protein coding genes that we've examined so far, and there's evidence of it occurring in all of the cancer types we've looked at so far. So, to recap, targeted somatic mutation is a somatic hypermutation-like process that occurs in all actively transcribed non-Ig protein coding genes in somatic tissues and results in new somatic mutations, and of course they're the ones that actually ultimately may result in a tumour. To sum it up then, TSM patterns are defined by the two concepts I gave you up front, and they are codon context and the motif that they're found on. And as I said, all protein coding genes undergoing transcription, in other words, the ones we need to produce new cells, and all cancers analysed. It's a very active system, and I should also say at this point that in a normal healthy cell replication process, there are many mechanisms that can repair an error if it occurs in our genes. It's when these systems fail due to either error or mutations in the genes themselves that are controlling the processes. The deaminases themselves occasionally take a hit and have mutations. It's when these errors occur that we actually end up having uncorrected mutations which accumulate in our system, and obviously the system ends up crippling itself because one error and the base excision repair not working properly, then maybe our ADARs will take a hit, and we end up with a very crippled genome which we can observe under a microscope and obviously at late stages in cancer. So again, a little bit of science. I just want to give the people that are visual in the audience just a recap on what's actually happening from the molecular cell level. First of all, we've got DNA to messenger RNA and DNA, the new DNA being produced. The first group of deaminases we talked about, the cytidine deaminases, they all need an open transcription bubble. In other words, single-stranded DNA to actually target the DNA or the motif that they want before they can actually introduce a mutation. That means, because it depends on a transcription bubble, it means that it only happens during transcription. It doesn't happen to other genes that are not undergoing transcription. And you see there a list of the deaminases that actually known to target single-stranded DNA. The next stage at messenger RNA level, there's ADARs 1, 2, 3 and 4. We know that they target double-stranded RNA. In other words, the bits that are joined together there in the middle and that form part of what we call a stem loop. We also now know, and there's still some controversy over this, the ApoBec1 and ApoBec3A, which is in the last 12 months, there's been a lot of work in this area, are cytidine deaminases that actually target RNA. That seems to be without question. But what is doubtable is whether it's double-stranded RNA or single-stranded RNA. But it looks like, in evolutionary terms, for a very long time, our cells and the deaminases have been an active part of enabling cells to change or adapt, if you like, by using the deaminases, even for single RNA entities, without even DNA being necessary, such as might have been in the early viral strains, and some of them that are still with us, of RNA viruses in particular, which is another story in itself. So summing that up, these are the deaminases we know exist at this point in time, 2016. This is where we believe that they act, their opportunity to act, and there are 15 of them. Most of those are tissue or tissue group specific. And you can see that if you look at it from the deaminase point of view, to introduce mutations from one generation of cells to the next, you can see that it is really essential that you have this messenger RNA intermediary and the reverse transcription step occurring. And it's unfortunate, but that's a model that's actually not 100% accepted yet. So we're all developing our knowledge, and I'm not sure what the alternative explanation is, but we'll see. TSM analytics. Just to describe the sort of tools I've had to get developed or work with, me and my colleagues. When I started, as I said, it was a manual process. The only resources I had was an Excel spreadsheet, a computer, public access to mutation databases, cancer mutation databases, and a copious supply of coffee, because it was a very slow and tedious process. In 2014, I was fortunate to partner up with some people from Melbourne, and HealthHack, I'm not sure if some of you have heard of HealthHack, which is where all the programmers get together in the health industry and knock themselves out over the weekend and have prizes for the best application. These guys got together and we developed a desktop application to actually facilitate the analysis that we needed to do and to speed things up enormously. And I'm still using that desktop application. Last year, we were really fortunate to receive a grant from the Victorian Government, the Tech Voucher Grant, which helped enormously for the development of a clinical grade test platform, a cloud-based one that's now housed by Amazon and has had several upgrades. Applied Precision Medicine did that development for us, and we're very pleased with what they've done. The core, we're keeping, in other words, the core processor is programmed in Python for the technical people in the audience, and we keep what's happened there as proprietary information because it's very difficult to actually patent a program. And around that, we've packaged a number of Oracle applications so that we can build, we can adjust our reporting, we can perform analytics and a whole heap of other functions that we need to perform. We also need to be auditable to get the standards approvals, etc. It needs to be a high integrity system. it needs to be rapidly scalable, and it needs to be adaptable to meet new standards because, as you know, the standards are changing all the time in this industry. The regulations we're working in and with today certainly might not exist tomorrow, so we need to be ready for that. And this is probably what I consider the most exciting part of the presentation this evening. We've finally got to the stage where we've got some early clinical trials active. In other words, the clinical proof and validity of what we're actually doing and what use is this for clinicians is really the question we're asking, and we've already got a number of partners. The first thing we're doing, and to do our homework properly, is clinical test platform validation. We've actually got that happening as a trial out at the Hudson at this point in time. We've also got a healthy population trial. No one knows what a healthy population, how many mutations are occurring due to these deaminases as we age. So we're doing that by age and by sex and amassing that data, and we'll use that as our baseline when we're comparing the profiles of individuals who may have cancer or some other immunodeficiency. We're also doing some cell line experiments. We're asking fundamental questions there as well. No one has compared the motifs for ADAR 1, 2, 3 and 4, yet those deaminases play an enormously important role in our central nervous system. We don't know what the future holds there, but we intend to find the motif and actually use whatever other information we can to find the association with central nervous system or disorders. We're also looking at viral strain diversity because deaminases, as you know, are the first line of defence when we get a virus. The virus mutates, tries to outsmart our immune system, and eventually there may be the adaptive immune system may come in and help us get over the virus. But these early stages, little is known about the collateral damage that these deaminases do to our own gene while we're actually fighting a virus, perhaps the flu, perhaps hepatitis. We're also in discussions for doing colorectal trials with the Royal Melbourne Hospital. We're looking at more data and more samples for the cancer progression associated signatures. And we're well on our way also with the healthy population trial. We're collecting samples and that's progressing well. It's received ethics approval, governance approval. The hepatitis B STOP trial, as we call it, has also received ethics approval and it involves 6 hospitals. That's a very active one. We want to know what a person who has hepatitis B, chronic hepatitis B, if they stop their medical regime, what is the outcome for that person? Will they recover or will they relapse? We don't know. We know the outcome, but we would like to be able to predict that before they cease their medication. And of course we're in discussion with others, but given that we're a small group, a new group, we believe we've taken on more than enough for year 1 of clinical trials. The following slides are just a few of the examples where we will be applying what we call our targeted somatic mutation profiling. In other words, looking at an individual tissue, saliva or bloods to see what's actually happening to their genes. In other words, what's the source of their mutations and what are the clinical implications. In scenario 1, you've got a chronic hepatitis person. This person may have contracted hepatitis at 28. They may need a particular medical regime and they have one of these profiles conducted every year to make sure that the medication, the efficacy of the medication is meeting their needs and they're not unnecessarily accumulating additional mutations due to, for example, ApoBec3G. At age 30, after they have a few normal tests, they suddenly see a spike in ApoBec3G. The market already has ApoBec3G blockers and antibodies, and some of the treatments for hepatitis involve the use of these. The virologists actually well understand the role of deaminases and the need to address those when they're looking at treatments as well. The second scenario is a case, and the last year 4 data is actual data from an autopsy sample. If a person, for example, is diagnosed with stage 4 melanoma, And they go on one of the new trials at the VCCC for Yervoy, one of the new immunotherapy drugs. And they come out and their oncologist says, well, they're in remission. In the future, we might be able to monitor those patients to say, well, yes, the deaminases are behaving as they ought to be. They're not accumulating unnecessarily additional mutations due to the collateral damage. So this is a scenario where after the 4th year, they stay in remission for 3 years, then suddenly in the 4th year, again, they get a big spike in the ApoBec3B. And we'd probably need to look for other information as well, because 3B is definitely associated with late-stage cancers from all of the samples we've looked at. ApoBec3B also undergoes a diversification in its deaminase binding domains. On that graph there, you'll also see that the ADARs, sometimes in late-stage cancers, we see in many samples where there's absolutely no new mutations due to ADARs. And that is actually associated with a very bad prognosis as well. And there's much more research to do in this area, obviously, but that's just 2 indicators we would be looking for in terms of making use of these diagnostics. The last scenario to look at is one where a young girl, might be 20 years old, goes in and has a BRCA1, BRCA2 test and finds out she's got the BRCA1, BRCA2 mutations. Her aunt has died of breast cancer. She goes to her oncologist and wonders, well, what do I do? Do I do an Angelina Jolie today, or do I wait until I have children? In this scenario, they would have a targeted somatic mutation profile, and it may be normal for a number of years. Then suddenly there may be a spike in AID deaminase activity, and it's already known that estrogen at some point in some women can trigger the levels of AID in their blood 20-fold. By those tests, we don't know if it's actually causing damage on the genome, though, because, as I said, healthy people ought to be able to repair those mutations if they're the result of that. So maybe the mutations are happening there, they're associated with AID. It's probably time to have a serious chat with her oncologist about what the next stop might be. Maybe she'll be lucky enough in the future to have blockers or antibodies to treat that and suppress the rate of mutation caused by, well, estrogen blockers is obviously one way of doing it and is a way that seems effective at the moment. Then you don't get the elevated AID. We may be able to more directly attack AID as well, but then we need AID as a first-line defence against viruses. So it's not, there won't be easy decisions. This is really additional companion data for oncologists and clinicians considering what the best care is for their patient. There's also, as I said, deaminases change. They change their deaminase binding domain at a certain point. Some of them predict cancer progression, some of them indicate cancer tumour suppression, and those that predict cancer progression would become potential druggable targets. And certainly we'd be looking towards the drug companies perhaps in 2017 to provide companion diagnostics to identify those new targets. So just recapping, TSM profiling methods are used to develop new clinical diagnostics and potentially develop new or identify new drug targets. And the key message here is that this would have to be done on an individual basis. In other words, personalised medicine, because of the differences among individuals. Your profile is unique. Your response to drugs will be unique, as we already know. So we're hoping for a future when we will be able to use such profiles to actually develop a personalised treatment regime rather than the one chemotherapy or the one operational procedure or radiation regime to treat all. We're very rapidly getting out of that space. So what it means for you, for me and for all our families is we're hoping to provide the diagnostics that will enable us to identify the right treatment to slow or prevent the accumulation of additional mutations. And now, and I'm aware that I've only got a couple of minutes left, these slides won't take long, we've got the innovation challenges. The biggest challenge for us has been funding. As I said, the government has been support, but this has basically been privately funded. I did take on board partners to progress the research, and hence the large range of projects we've now got underway and clinical trials. We've taken on board partners, and I thank them for coming along tonight. There's also the challenge of intellectual property. Intellectual property is a hard game to play sometimes. It can be challenged. The international rules are different, and it costs. It costs a lot of money. And we've certainly been patenting. I took out my initial patent application in 2012 when no one knew what I was talking about, and I had great difficulty finding an immunologist who could understand me to get those first patents drafted and submitted to PCT stage. The collaboration, to take basic research through to development of diagnostic assays, it actually requires a lot of people, not just a lot of money, but a lot of capital resource, a lot of people resources as well, human resources. It needs collaboration with contract research organisations, clinical trial nurses, patent attorneys, regulatory advisors, people to actually implement our standards and documentation and keep us in, so that we can actually get the certifications we require. All of that happens in a market undergoing rapid change. Almost daily you can see takeovers. There's a lot of movement internationally and globally in this area. There are also a lot of new companies developing with fantastic claims about what they're going to do with their new genomic diagnostics. And obviously we have to keep an eye on those. How are we going to earn revenue? We've got a platform technology essentially, so it's basically a licensing play. We're hoping to partner with some drug companies, or at least one next year, to start that, the money flow, the cash flow, if you like, working with immunotherapy drugs and perhaps some others. The good thing about having a platform technology is, one, that it isn't just limited to oncology. The deaminases play a role and are now being implicated in a range of other diseases. So there's geriatrics, diabetes, transplant biology, immune-related autism, and veterinary applications. So just to sum up then, the codon context, which I indicated initially, and the motive have provided us with concepts to develop the tools, the analytic tools, that will be able to identify the source or the likely source of mutations in any genome that we analyse. This enables us to develop new diagnostic tools. And of course, going from basic research, which we've done, right through to developing new diagnostic tools, there's a lot of innovation challenges, and it's a journey. And we're just really at the very early stage of clinical trials, so we have a long way to go and a lot of work to do. Thank you.